Okay, this is the Professor, CBMD PhD, and today's Friday, June 30th, 2023. It is 12:58 and 10 seconds, a.m. Eastern Standard Time, and today's research, cancer invasion. So let's hop to it here. Researchers identify mechanism of cancer invasion, date June 27, 2023. Source, Penn State. A cancerous tumor is the accumulation of cells uncontrollably dividing, some of which can invade other parts of the body. The process is difficult to predict in detail, and eradicating the cells poses even greater difficulty. Now our research team has revealed how the excess initiates, shedding light on a potential therapeutic target to halt the invasion and providing a prognostic marker to help clinicians select the best treatment option. As cancer cells randomly detach from the primary tumor and disseminate everywhere, they often exhibit coordination and collaboration. A leader cell may emerge to coordinate the invasion, thereby enhancing the efficiency of cancer cell dissemination. In this study, they found a molecular marker for leader cells that enables them to predict the invasiveness of the tumor and how they invade. In cancer cells derived from human patients with muscle-invasive bladder cancer, the researchers designed a nanobiosensor to track the non-coding RNA, which refers to the extensive lengths of genetic material that do not encode genes but regulate how a cell expresses them as proteins. Non-coding RNA are often referred to as the dark matter of the cell. While many RNA are involved in protein expression, non-coding RNA do not encode proteins, as their functions and how they regulate cell processes are still poorly understood. They developed the sensor to study non-coding RNA and their potential contribution to cancer progression. The sensor enables the researchers to identify and track individual non-coding RNA molecules of interest as they typically behave and function in cells. In conventional analysis approaches, researchers usually cannot study how they function in space and time because the cells are typically fixed or broken apart. Using the sensors, the researchers monitored how much and where non-coding RNA was distributed in the cells during collective cancer invasion. They found that MALT1, a gene associated with metastasis in lung, bladder, and other cancers, was highly present in leader cells. Importantly, researchers said MALAT1 expression increased when a cancer cell became a leader and decreased when the leader cell was no longer needed, such as when the invasion process ceased or when it was replaced by another leader cell. They also found that reducing the expression of MALAT1 in cells prevents the formation of leader cells and abolishes the invasion of cancer cells. Overall, the single-cell analysis suggests that MALAT1 plays an essential role in regulating leader cells during collective cancer invasion. If they can comprehend the crucial characteristics and functions of leader cells, they might be able to help clinicians identify aggressive disease and predict the behavior. They hope this study will lead to the development of prognostics and therapeutic approaches targeting bladder and other cancers. As, for example, if applied clinically, determining the presence of leader cells in aggressive disease could enhance a physician's understanding of an individual patient's prognosis and inform the most suitable treatment strategy. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.